waving?
I see you waving, but I can't hear you, buddy. I'm so
I got
sorry.
a question.
Yeah, I can't hear you.
Look at the chat. I'm finna text you.
Yeah.
He says.
Anyway, yeah. So the first word for attendance is more, and let's jump into it. Let me pull up the PowerPoint. Again, we're going to finish up chapter 10. Oh, yeah, another thing for attendance. Some of you guys may have noticed, well, two things, actually. I haven't posted the video yet for Friday. So if you're wondering where that is, you haven't lost your mind. I just haven't posted it yet. I actually decided to spend time with my family this weekend. So I apologize. But, yeah, I'll get it out to you as soon as I can. And another thing for attendance, if you were to look at your public grade sheet and the attendance section, there was a little message that said the first person who sees this will get 25 points of independent or of extra credit. And Skylar Simpson won. She was the first person to see it, so she got the 25 points of extra credit. If you're wondering what the purpose of that was, it's because you guys should be on top of your grades. So I'm rewarding you for checking in on things like that. Anyway, where are we at here? Where are we at? Mutations. Yep, this is where we left off. We were talking about mutations, specifically the one that causes sickle cell. Anyway, really quickly to back up, when we're talking about mutations, we are talking about a change in your genes, not the ones you wear, but your genes with a G, right? The codes, the A's, the C's, the T's, and the G's. And before we specifically talked about the sickle cell example, I said in general, those changes can be really small, like this example where we just changed a T for an A. Or it could be a big one. You could have a whole chunk of DNA, like a whole chunk of chromosome that's messed up, right? It could be small, it could be big, it can be anywhere in between. Again, your book gives the example of sickle cell. I'm not going to ask you any questions about sickle cell on the exam. This is just an example. So when you're studying, don't feel like you need to memorize the fact that you went from a T to an A and you went from a GLU to a VAL amino acid in the sickle cell mutation. Nothing to worry about there.
The first word was more.
And I will say this, too. I meant to say this on Friday before we even started talking about mutations, because Friday we finished up talking about this whole process right here where we go from, DNA to RNA to protein. And even though we're about to talk about how viruses work after we talk about mutations, I do want to say this very quickly because I didn't say this on Friday. People talk about gene therapy a lot. Gene therapy is when you go in and change your DNA, right? That's your original code. So we're using the analogy that I was using last week of, we have this set of instructions. This is the room. And we have a set of instructions on how to build stuff on the campus, including instructions on how to build a bench. That's the analogy I was using. So gene therapy would be like going in and changing that one book, right? You go in there and change your master copy. That's gene therapy. And the reason I say this is because I see a bunch of idiots. I see a bunch of dumb stuff on the internet being passed along about the RNA vaccine for COVID. We don't have time to really get into that vaccine, but I will say this. It's not gene therapy, and it cannot mess up your DNA. Because remember, the process goes from DNA to RNA, and then from RNA to protein. So if you're getting an RNA vaccine, it's not going to affect your DNA because that's the other direction, right? You're getting an RNA vaccine. They're putting mRNA in you, and that is being translated into certain proteins. Just in case I'm already talking about it, so go ahead and tell you. Specifically, there's proteins on the outside of the COVID-19 virus, right? The proteins themselves aren't dangerous. It's the stuff that's on the inside, as you're going to learn about a little bit later. That's the dangerous part. So all they're doing is giving you the protein that's on the outside. Your body makes it, and then it recognizes it. So whenever you do get exposed to the virus, hopefully your body's like, oh, yeah, I know how to fight this because I've already been exposed. But anyway, I wanted to point that out. Now let's go back to mutations. Small, big, anywhere in between. We talked about the one for hemoglobin. This is the exact slide we left off on. Here's some stuff you need to know. There are two categories of mutations. There's substitutions and there's deletions / insertions. The good news about this is this isn't one of those situations where you have really weird names like you get in biology, right? The name really tells what happens. Like substitution. What do you think substitution means? Anybody want to guess what a substitution is before I move forward? What are we substituting? Of course, if you're online, I can't hear you. That stinks, and sorry. But for those of you in person, anybody want to guess what a substitution might be? What are we substituting? No guesses? All right. Hopefully this makes sense. Actually, the example I just gave you that we didn't really talk about too much today, that we talked about on Friday, that was a substitution. That was a T. Instead, it was substituted for an A. We are substituting one base for another, right? One nucleotide for another. That's all a substitution is. It's a substituting one base for another. And here's a different example. This is from your book. Up top here, you have the normal gene, right? Which then gives you, that's mRNA. So that's the normal mRNA followed by the normal chain of amino acids, i.e. polypeptide. And then here at the bottom, you have a mutation. So instead of having a G, like you're supposed to have, there was a mutation, and it substituted it for an A. And because of that, instead of having this GLY amino acid, you have an SER amino acid. And again, like I said on Friday when we were talking about sickle cell, that might seem like a small change and no big deal, right? It's like, oh, you've got one amino acid for the other. But keep in mind, a protein's function is based on its shape. It's very important. So if you change that one amino acid, then you change the chemistry of it. Because like I said on Friday, some amino acids are polar, some are non-polar. Some are hydrophobic, some are hydrophilic, some are charged, some are not charged, some are positively charged, some are negatively charged, right? All these things that make them, and they all have different shapes and lengths, right? So they all interact differently. So by changing that one amino acid, you're changing the shape of the entire protein, and therefore you're changing its function. All right, here's the next one. This is not tough. This is a deletion. And guess what happens in a deletion? You delete one of the nucleotides, right? So you can see here, again, at the top is the normal mRNA and the normal polypeptide. Here you're supposed to have a U, and then here obviously something went wrong and the U was not included. Now this is a bigger deal than a substitution. Look, when you get rid of this U, boop, right, you plop it out, everything shifts to the left. Everything shifts to the left. And remember, proteins are actually a lot longer than this. This is just a simplified version. They're really long. So if you have this happen, everything to the right that you can't see, hundreds, maybe thousands of amino acids, everything is messed up because everything just shifts one to the left. So now every polypeptide, every amino acid downstream is going to be messed up. So you talk about a big difference. That's a huge difference. So obviously, deletions are a lot worse than substitutions. And of course, the same can be said for insertions. So here, you're supposed to have U, and then a GGC. But what happens instead is you put this G right between these two U's, right? So now you have UUG. And again, you've messed up everything downstream, even though we can't see it in this picture. Remember, there's hundreds, maybe thousands of amino acids downstream. And it's all messed up. Pretty simple stuff, right? What you need to know for the exam, what's a substitution? You're substituting one base for another, one nucleotide for another. What's an insertion? You're inserting an extra nucleotide. What's a deletion? You're deleting one of the nucleotides. Which ones are worse? The insertions and the deletions are equally bad. They're worse than the substitutions because, again, once you do that, you change everything. And there's a word for that you're about to learn. I'll come back to that. Let's say the next word for attendance. I'm going to circle it and not say it, as I do sometimes. Right there it is. Anyway, another term you need to know for the exam is missense. That is a mutation that changes the amino acid coding. So far, all the examples I've given you have been missense, because whether it been an insertion or a deletion or a substitution, everything I showed you, it ended up where you went from one amino acid, the proper one, to a different new amino acid, which was not the one that was supposed to be there, right? That is a missense mutation. You should know that for the exam. The opposite of that would be a nonsense mutation. I wish I had the code. Do I? I don't. Try, I should put that back on the PowerPoint. Try in your mind to remember that big chart I showed you, the big circle, where I was showing you how to translate stuff. Remember, like you would start here, like, okay, there's an A, and then a U, and then a G. Remember? And it would tell you what amino acid it is. If you can remember that, the thing about amino acids is there's usually more than one codon for a particular amino acid. So in some cases, and again, we're talking about the nonsense here, in some cases you can change the base, right? You can change the letter, but the word still says the same thing. The word still says, bring me a tryptophan or whatever amino acid it is, right? So that's a nonsense mutation. So yes, you did change the nucleotide, but, excuse me, good grief, I'm out of it today. We're talking about nonsense. So we're talking about changing it to a stop codon. I'm so sorry. Good grief. I apologize for that. Nonsense is changing it to the stop codon. My brain was already somewhere else. I'm moving ahead. So are there any questions? Okay. So one of the ones we've already talked about actually has a name to it. We already talked about how if you insert something or delete something, it's disastrous because you move the whole thing, everything downstream. And that is what we call a frame shift mutation.
Anybody know
more shifting the frame? Yeah, and it's like we said, we said earlier, it's usually very bad.
More.
The book way of saying it's usually very bad is it usually produces a non-functioning polypeptide.
Yeah.
Again,
M-O-R-E.
for the purposes of this conversation, polypeptide is pretty much the same thing as a protein, right? It's a non-functioning protein. You really messed things up. Let me show you this picture of this before I see if you have any questions. This is not a picture from your book, but it's a great example of a frame shift mutation. The reason I like this one better than your book is because even though it's still small, it's bigger than the one in your book because we have 1, 2, 3, 4, 5, 6, 7. At least this one shows 7 amino acids. So anyway, because of this frame shift mutation, you can even see which one it is. Everything is shifted. That's what we call a frame shift, right? So what this should be read, the CAT and then TCA, CAC. But what happens is something happened downstream that way, and it caused everything to move over one. So what should have been a CAT is now an ATT. And what should have been a TCA is now a CAC, right? So when you have a frame shift mutation, everything shifts, and it's really bad. And that's pretty much all you need to know about that. So are there any questions about missense or nonsense or frame shift mutations? All right. So here's a little table from your book that says that kind of sums up everything we talked about, except this one, which I erroneously was trying to talk about earlier. Silent mutations. Those are the ones where you substitute one base for another, right? One nucleotide for another. So you change the letter, but you didn't change the code. The code says, bring me this amino acid, whatever this amino acid is. That's the one I was trying to talk about earlier. So silent, missense, nonsense, frame shift. And you should know these for the exam. Any questions? All right. I cannot speak. Mutations occur in a number of ways. I'm going to talk about this very quickly because you should already know this in general. Basically, bad stuff can cause mutations, right? This is where you get cancer a lot of times. So physical mutagens like X-rays, UV light, right? That's why you're supposed to wear sunblock, because the sun has a lot of UV light and it can literally mutate your DNA and your skin cells, and that's where you get skin cancer. Nuclear bombs, that'll do it to you. Then there's also chemical mutagens like smoking, right? Smoking is bad for you. That's one of the reasons why. The only thing you really need to know, if anything, from this slide is what a mutagen is. A mutagen is just something that causes mutations. You don't need to know any of the examples. Just know that a mutagen is something that causes mutations. It's a good independent work topic if you want to look it up. Just write about a bunch of mutagens. What are the things? I mean, there's a bunch. You couldn't list them all. I mean, you could, but I wouldn't expect you to. But you just go and find some things that are bad for your DNA. Things that causes mutation. Any questions about this? Trying to move quickly here. All right. Often, as you might imagine, mutations are bad, right? They can be very harmful. Obviously, if you change the code and you change the shape of the protein and you change the shape of the protein, you change its function, that's usually very bad. We talked about that when we talked about the sickle cell, right? Because the purpose of that protein is to carry oxygen. So if you mess that up, it doesn't do such a good job of carrying oxygen. And guess what? We need oxygen to live, as you know from chapter 6 when we talked about cellular respiration. Anyway, sometimes, though, these mutations can be good. And keep that in the back of your mind, because that's what we're going to talk about when we jump into natural selection, which is actually going to be today for chapter 13. And you guys have already had that lab, right? So in that case, that light-colored moth that had the mutation that caused it to have dark-colored offspring, that was a good mutation in that case, because it just so happened that things were really polluted at the time. Who knows? It's possible. I don't know how probable it is, but it's possible that prior to the Industrial Revolution, maybe there was a moth that had that mutation before and popped out a bunch of dark-colored offspring. But because there was no pollution, maybe those dark-colored offspring stuck out like sore thumbs and they were just eaten right away, and that was the end of that, right? Yes, there was a mutation in the gene pool, but then everything that had it was killed and eaten, so it was not passed along, right? Anyway, yeah, that's it. So any questions about this slide? All right, here we go. The fun stuff: viruses and other non-cellular infectious agents. You're not going to be an expert on viruses after this, but you're going to know a little bit more than you used to, and that's a good start, right? So if you ever get curious about COVID-19 or the next pandemic or whatever, you're not going to be starting from scratch. You're going to have a little bit of a foundation to work with. Let's get into it. What is a virus? Well, it shares characteristics of living organisms. Like it has genetic material in the form of nucleic acids packaged in highly organized structures. So there are some similarities, but it's not considered alive. Why is it not considered alive? Well, it's missing two of the life properties. First of all, it's not cellular, right? Yes, it looks a little bit cellular, right? Like it says right there, it's highly organized, it's structured, it has order, but it's not a cell. And it cannot reproduce on its own. That's where we come in, right? That's what viruses do. All they want to do is reproduce. They're basically out there hijacking cells so the cells can do the reproduction for them. Any questions about this slide? All right. A virus, as your book puts it, is an infectious particle consisting of genes in a box. It's some form of nucleic acid wrapped in a protein coat. It could be DNA, it could be RNA. We're going to talk about both of those here shortly. In some cases, it is enveloped in a membrane. So sometimes it's just protein. Sometimes it's the nucleic acid in some sort of protein, like a box of protein, if you will. Sometimes that itself has a membrane, just like our cells. And a lot of times that's actually how it helps infect us, because its membrane merges with our membrane, and that's how that works. Like I already said, viruses cannot reproduce on their own. It's a very important concept. Maybe that's, I don't know, that might be a test question. It's just so easy. I don't know. I might not put it on there. Anyway, because it can't reproduce on its own, that's why it has to infect other living cells. Right? So we know now, because we've talked about transcription and translation, you know that you've got little cellular machines, right? All these enzymes, the ribosomes, the tRNA, right? You know that the mRNA attaches to the ribosomes, and then the tRNA is like, all right, here's this amino acid, here's that amino acid, and it goes and it builds a polypeptide. So viruses basically just take advantage of that. It's like, all right, well, you've got the machines to read mRNA, here's some mRNA. It would be like if you didn't have your own 3D printer, but you wanted to 3D print something, so you sneak into somebody's house who has a 3D printer, and you're like, boop, you put in the code, you print what you need, and then you take it and you leave, right? It's similar to that. Here's a picture, just one version of it. There's so many different versions of viruses, but this is the one I was telling you about, where there's a protein coat on the outside and a nucleic acid on the inside. In this case, the nucleic acid happens to be DNA and not RNA, but there you go. And of course, sometimes they have a membrane on the outside, but that one's just one with a protein coat. It's just an example. You don't need to know anything about that for the exam. All right, here we go. We're going to talk about different types of viruses. The first one is called bacteriophages, or some people say bacteriophage. Anybody want to guess what kind of organisms that those viruses attack? Right here. Any guesses? Sorry for you, those of you online, I can't hear you. No one wants to guess what kind of organisms bacteriophages attack. Doesn't hurt to be wrong, and if you're right, you'll get extra credit. No guesses? I'm so disappointed. Yes, thank you. Yeah, I'm hoping you guys were just thinking that surely it's not that easy. Yes, it's bacteria. It's in the name, right? They attack bacteria. When you send me an email for attendance today, can you remind me that you got that right, so I give you some extra credit. Yes, bacteriophages, they attack bacteria. That's where the bacteria part comes in. The phage or the phage, that kind of just means eat, right? Eaters. So, which is interesting because I don't really eat them, and you'll see why here in a second. But anyway, as far as the exam is concerned, that's about all you need to know about bacteriophages, is that they attack bacteria. But I'm going to teach you a little bit about them. They are DNA vaccine. I want to say vaccine. They are DNA viruses, not RNA viruses. Meaning, well, you'll see here in a second, but that's what they look like. That's a picture of one of them right there. That weird looking thing looks like a spacecraft or something. It's actually a virus. There's a scanning electron microscope picture of a few of them. So here's the bacteria cell right here. 3 of these things have attached. And what they do is they kind of basically just like a syringe, they just insert, and then they put in the DNA, and then their DNA gets incorporated into the bacteria DNA, and it's pretty much there forever. There's another picture. So yes, there's different types of bacteriophages. This one is called a phage lambda, and obviously there's different types of bacteria. This particular type of bacteria is called E. coli. But yes, this is what I was talking about. So it inserts this DNA, that's the red stuff, and then you can see the green stuff is the original bacteria DNA. So what happens is this incorporates itself into the bacteria's DNA. So then every time this gets transcribed, then that mRNA gets translated into all the different protein parts that make up a virus, and then the cell builds a bunch of viruses until there's so many that they can't hold it anymore, and it busts out, and then those viruses escape, and then they go on to infect more E. coli cells. So, any questions about this slide? All right. I'm going to go through this quickly. I'm not quite sure I'm going to ask you anything about it because, again, these things attack bacteria, which means they're really not that important for you. But anyway, if we're talking about these things that attack bacteria, there's 2 different cycles. There's the lytic cycle and there's the lysogenic cycle. What I love about this is the word lytic is shorter than the word lysogenic. And that way you can remember that the lytic cycle is the shorter of the 2 and the lysogenic cycle is the longer of the 2. Let me just show you the picture. It's actually pretty simple. Well, the way you learn it is pretty simple. Basically, the lytic cycle is straightforward. It's the one I just showed you. The phage attacks, it inserts its DNA. That DNA incorporates itself into the original DNA. The cellular machines do their business, right? They read the code, they make the mRNA, and then they make the protein, and then they assemble all these parts for the new viruses, and then it explodes because there's so many viruses it just can't handle it anymore, and then those things go out to infect a new cell, and the cycle continues. That's it, right? That's the lytic cycle. Very straightforward. It gets a little bit more complicated when we do the lysogenic cycle. And the lysogenic cycle, what happens is that DNA, that viral DNA, gets incorporated into the bacteria DNA, but then the code's not read, right? There is no transcription and translation. So what's there? That viral DNA is just sitting there. And every time this thing goes through mitosis and reproduces itself, it does a perfect copy. So it includes the copy of that viral DNA. So then you get hundreds, thousands, millions, whatever, millions of cells that now have that viral DNA in it. That's the lysogenic cycle. That's what I mean by it's longer, because it keeps going, right? Every time that thing reproduces, that code, that original viral DNA, it gets copied as well. As a matter of fact, we use this to our advantage in medical technology, biotechnology. We sometimes use viruses to put in our own codes. Like if we want to amplify a certain code, right, we can put it in bacteria and then have the bacteria do it for us. It's like little copying machines, and we do that here at WVSU. That's actually called cloning. So most people, when they think of cloning, they think of, you take a sheep and make a new sheep. That is also a type of cloning, but this is the type of cloning we do all the time. But anyway, so that's part of the lysogenic cycle. Again, the big difference here is instead of the viral particles being made right away, the viral DNA is incorporated into the genome and it just sits there dormant. Of course, eventually that dormancy is lost and it does become activated, and then it goes into the lytic cycle, which is where the viral particles are made, and then the viruses are put together, and of course it explodes and reinfects. So again, you should remember the lytic cycle is shorter, lysogenic cycle is longer. If you can remember that, hopefully you can remember the reason why the lysogenic cycle is longer than the lytic cycle is because the lytic cycle is just quick death, right? Here's the code to make the protein or the virus, and it makes the virus right away, and that ends the life, right? Here, longer because that code is incorporated into the DNA of the bacteria, and it just keeps copying it. So, any questions about this slide? So again, if you're studying, don't study this like you have to know every little step to these cycles. Just know that the lytic is shorter than the lysogenic. All right, the next thing we're going to talk about are plant viruses. So I'm sorry, anybody want to guess what kind of organisms that these viruses attack? Plants, yes. Sorry, I'm not giving extra credit for that one just because the first one I can see is a little bit tricky because you're probably like, can't be that easy. But now you know it is indeed that easy. So yeah, plant viruses, I'm going to go through these pretty quickly too because unless you're into agriculture or, I don't know, growing plants, this probably doesn't directly affect you too much. It definitely does in other ways, because you eat food, you eat plants. So anyway, plant viruses. They're not usually deadly to plants. They usually just stunt plant growth and give them problems. They diminish crop yields, which again is bad if you're, it's bad no matter what, whether you're growing them and selling them or if you're buying them because prices will go up and not just for inflation. Anyway, most of them are RNA. That would be another independent work topic. Look up some of these. What are some? Give me some plant viruses because I don't only give you one example, so you can find some more. Find some that are DNA for me. Anyway, the example that your book talks about is the tobacco mosaic virus. It's a rod-shaped spot with spiral arrangements of proteins surrounding nucleic acid. This is what it looks like up here. That's what I mean by rod-shaped. I know it's a little bit hard to see, but that's a spiral of protein. It infects tobacco and related plants. It causes discolored spots on leaves. And I think the reason your book, you chose this one as the example is because this is the first virus that was ever discovered. You don't need to know any of this for the exam. Maybe I'll ask you, you might need to know that if we're talking about plant viruses, it's not deadly and it's usually RNA. But again, I'm probably not going to ask you that. Speaking of plants, the next word for attendance is going to be pine, like a pine tree. Pine. Any questions? Wait, hold on. There's a little bit more about plant viruses before I take any questions. That's what it looks like, right? That's a tobacco leaf. It's not supposed to have those colors. It has the tobacco mosaic virus. Here's a picture of it. Again, it just looks like a rod. But if we zoom into this part right here, you can see the RNA is spiraled around inside, and then there's a protein spiral on the outside. That's it. Any questions about plant viruses? All right. Oh, sorry. I want to talk about more. Again, I teach out of the book. This is in your book, so I included it. Obviously, for a plant to get infected, they don't necessarily have a respiration system, even though they do cellular respiration. But anyway, because of that, the virus has to get past the plant epidermis. And for that reason, a damaged plant is more likely to be infected. Sometimes insects carry the virus. Sometimes farming tools can spread viruses. Like it's almost like surgery. What you're supposed to do when you're working on trees and things like that, you're supposed to clean the blade and make sure they're clean. Otherwise, you might be spreading viruses. There's no cure for most viral plant diseases, which is, sure, they're not deadly, but there's also no cure. And because of this, agricultural scientists focus on prevention and breeding or genetically engineering varieties of crop plants that resist viral infection. So there it is. Big bad GMO, right? Everybody's afraid of GMO. Sometimes the GMO just might be looking at the genome of a strawberry plant, I don't know, and saying, okay, this particular gene makes this strawberry susceptible to this virus. So let's just cut out that gene, right? And then there you go. You have GMO. And a lot of people are afraid of it because they don't understand it. Anyway, talk about that some other time too. Any questions about plant viruses now that we're done with that? All right. Notice, too, that we're only talking about plants, bacteria, and animals. Nothing about protists and nothing about fungi. So if you want, you can look that up for independent work, too. What about viruses that attack fungi and protists? Anyway, animal viruses. This one's a little bit more important because guess what? We are animals. Obviously, animal viruses are common causes of disease. I don't know if you guys have heard of it. There's a virus that causes something called COVID-19. It's been up and coming. So I'm sure you guys know about that. There's a lot of examples. Obviously, we're living in a pandemic right now. This book was written before COVID-19. So at the time, the deadliest virus ever was the flu virus. And it still might be. Now, don't get me wrong. In the past 2 years, I'm sure COVID-19 or the virus that causes COVID-19 has killed more people than flu, but flu's been around longer, right? So flu, it's got bigger numbers for that reason. It's been killing people for a long time. Anyway, so since your book talks about the flu virus, so will I. Like many animal viruses, the flu virus has an outer envelope of a phospholipid membrane, which is just basically telling you it kind of has the same membrane as a cell membrane, which is basically how it incorporates the cells. It also has spikes of protein. Now, these two things that I circled, if we're just looking at that, I might as well be describing the virus that causes COVID-19 because it also has a membrane. It also has spikes. Like I was mentioning earlier about the vaccine, that's how it works. It gives you the code to make the spike protein, which is harmless, and then your body can recognize it. Anyway, that's just an example. I'm not going to ask you any questions about flu. If anything, you might need to know that it's generally considered the deadliest virus in history, but probably not even that. So any questions? 8:32. We might not jump into chapter 13 today. At least that would make things easier. Anyway, if we're talking about animal viruses, which we are, a lot of them have RNA instead of DNA, right? So again, like I said earlier, there's different types. There's RNA viruses, there's DNA viruses. Obviously, if we're talking about RNA viruses, well, anybody want to guess? Let's give you the multiple choice. Because remember, we've talked about 3 different types of RNA. Let me think about this alphabetically. There's mRNA, there's rRNA, and there's tRNA. So this is good. This is not trivial. This is something you will need to know for the exam. Not necessarily about the virus, but if you had to guess, what kind of RNA do you think we're talking about here? No guesses, that's okay. It's mRNA, because remember, mRNA is the messenger RNA. That's the stuff that's the code. That's the code that says, all right, bring me this amino acid, bring me that amino acid, bring me this amino acid, right? That is the code on how to build a protein. So if you're a virus and you just want to get proteins built, that's the mRNA, that's the RNA you're working with. Like here's some mRNA, read the code, build me some proteins, and assemble me some new viruses. So yeah. Anyway, your book also gives some examples of which ones are RNA viruses. Flu, common cold, which is the one that's related to the virus that causes COVID-19, by the way. Measles, mumps, AIDS, polio, these are all RNA viruses. You don't need to know that for the exam. I'm not going to ask you which ones are RNA, which ones are DNA, but now you know. Speaking of which, here's some examples of the DNA viruses. Hepatitis, chickenpox, herpes, the gift that keeps on giving. And you'll see why here in a second, because we're going to talk about the DNA virus. This should look familiar, sort of. So I forget which particular virus, which particular type of virus this is supposed to show, but there you go. You got the membranous envelope with the spike proteins on the outside, right? Because that's what allows it to get into our cells. And then here, this is interesting to me. This looks like a bunch of plant viruses, at least the one I just showed you, because it's a rod spiral of RNA inside the spiral of a protein coat, right? Except instead of just going directly in like that, you've got the membrane on the outside. Anyway, I'm going to move forward. So you're not have any questions about that. All right, let's talk about how this works. This is just generally how it works. Obviously, each virus is a little bit different, but what we're doing is trying to give you the overall scheme of things. So what we have here is an RNA virus. Again, it's got the membranous outside. So what it's done is kind of merged with the cell membrane. That's what's going on up there. And then all this stuff comes in, right? That RNA comes in. It's wrapped up in proteins, but the proteins become uncoated right here, right? You can see these proteins going all over the place, opening up so we can get to the RNA itself, right? That's the mRNA. That's the code that says, all right, use this code and build me some proteins, right? That's basically what happens. So not only does it need proteins, right, because you need to build the virus, but obviously you also need some more copies of the genome itself. You need to copy that mRNA because once you have the proteins to make the virus, the new virus, then you also need to put the new mRNA in there. So that's basically it. I'm not going to ask you all those details, but now you know, right? Now you know how generally how a virus works. They break in, they take over your cellular machinery. The cellular machinery copies the genome and follows the instructions on how to build the proteins, and then reassembles all that stuff. Like, all right, we've got the new proteins, we've got our new viral DNA, or excuse me, viral RNA in this picture. So we put it all together and make a new protein, and then boom, it leaves the cell, and the cycle continues. Any questions about this? All right. Yeah. So this is everything we just talked about, except one big picture. So there it is again. Yeah, nothing to say there. If you download the PowerPoint, you can watch this animation that shows it in action. Here's a picture of it happening. This is happens to be, excuse me, this is not a picture of it happening. This is a picture of the mumps virus. You can see the little protein spikes sticking out. You can see an envelope. I don't know why. I haven't thought about this until just now, but I should probably update this picture and
give you
guys a picture of the virus that causes COVID-19. That will be a little bit more relevant. Anyway, not all viruses are reproduced in the cytoplasm. So what I just showed you happened in the cytoplasm, which makes sense. You should have known that without me asking or saying it, because what we were talking about was translation, right? And we know that translation is taking the mRNA and turning it into protein. And you should know for the exam that translation happens in the cytoplasm, not in the nucleus, right? That being said, again, it doesn't always happen in the cytoplasm. Your book gives an example, the herpes virus, which is also causes chickenpox, shingles, cold sores, genital herpes. It's very versatile, but it's a little bit trickier. It is actually a DNA virus, and that makes it tricky because it reproduces in the cell's nucleus. And not only that, but it incorporates itself into your genome. So it is a little bit like the lysogenic cycle that we talked about in a sense, because it can lay dormant. I don't know if any of you have herpes, or like me, I've had chickenpox. And because of that, like that just lies dormant. And then if there's enough stress or something triggers it, then you can get shingles. So I've had shingles, which is, I guess, not normal for somebody relatively young. But I had it, and that's what happened, right? I had this virus that was lying dormant in my genome. Something triggered it, and that's where the shingles came from. Anyway, probably not going to ask you anything about this, but there it is anyway. Any questions about this slide? All right. We're still talking about herpes here. It can, well, yeah, it's basically what I said previously, except now we're getting a little bit more specific. When I say it incorporates itself into your genome, your book gets pretty specific here and says that the DNA from the herpes virus will remain behind in the nuclei of certain nerve cells. And like I said, it remains dormant until something, some stress makes it come back. There's obviously unpleasant symptoms. Yeah, and that's also, yeah, like I was saying, the personal example I was giving was shingles. But I'm sure if you or anybody you know has herpes, especially like oral herpes, you might know that, like, there's all of a sudden, you know, they're fine, and then all of a sudden, boom, they've got an outbreak, they've got a sore. And that's what happens, right? Something triggered it. Anyway, be specific with numbers here. Over 75% of adults have herpes simplex 1, which is like oral herpes, the cold sores. And over 20% have herpes simplex 2, which is genital herpes. So I'll put a big old X to that. You don't need to know that for the exam. Any questions? All right. Your next word for attendance will be genital herpes. It's right there. All right. So what makes a virus deadly or bad or good? How
Did
bad
he
are
give
they?
a third word?
Well, the damage depends partially on how quickly the immune system responds and the ability of the tissue to repair itself. So, for example, if you're attacking a tissue that's really good at repairing itself, that's generally not as bad. Versus if you attack something like the polio virus, where it attacks your nerve cells, which don't really do a good job of replacing themselves, right? That's a little bit more serious. That's why the common cold might not be as bad as the polio virus. Of course, a lot of people don't think anything of the polio virus because thanks to vaccines, it's essentially been eradicated, but it is a really bad virus. Yeah, those are just examples. I'm not going to ask you anything about that. If anything, just know the main bullet point here, right? That not all viruses are created equal. Some are worse than others, and the thing that generally the two things that make them either worse or better is what it attacks and how quickly the immune system can respond. And again, that's what vaccines are for. Because if you get COVID-19, your immune system should, and obviously there's a lot of people who died, so it didn't work for everybody, but your immune system will learn how to fight it, right? And eventually you'll get rid of the infection. But that's the whole purpose of a vaccine is like, hey, it gives you the safe part, and then your body learns how to fight it before you even get exposed to it. So that way, when you do get exposed to it, it's like, all right, I got this, right? Your immune system knows how to do it. As always, your book has this, The Process of Science. As always, I'm going to skip it. It will not be on the exam. There might be questions on the study guide. I don't remember, but it will not be on the exam. I still highly recommend reading it and thinking about it because it's a good way to think. Helps you think scientifically. And if you have any questions after reading it, let me know. Moving forward. Here we go. Almost done. I'm sure you guys have all heard of HIV, which is the virus that causes AIDS. Let's talk a little bit more about it because, it's still not exactly cured. It's one of those things that's been around for a while. It's plaguing us. Let's talk about what makes it so bad. AIDS, that stands for Acquired Immunodeficiency Syndrome. Basically meaning the thing destroys your immune system. So it's bad. It's not just that you have AIDS, but then you basically have no immune system. So you get every other disease that you're exposed to. It is a retrovirus, and you do need to know what a retrovirus is. It's a little bit nastier than the ones we've talked about before. First of all, it's an RNA virus. So obviously it can either be RNA or DNA. Now you know it's an RNA virus. But it reproduces with a DNA molecule, which should be very interesting if you're actually paying attention and you remember everything I've said. And I don't blame you if you don't, because I'm kind of going quickly. But it is the reverse of the usual DNA to RNA to protein flow, right? So far, everything I've told you is transcription and then translation. Or in viruses, if it's a RNA virus, then it's just straight up translation, right? This makes things go backwards. But guess what? For it to happen, you have to have a particular enzyme, and you need to know the name of this enzyme. If we're talking about retroviruses, you need to know reverse transcriptase. Know that for the exam. But guess what? It's in the name. What does reverse transcriptase do? It does the reverse of transcription. So instead of going from DNA to RNA, which is what transcription is, it does the opposite. Also, mind you, that this is part of the AIDS virus. You don't just have this hanging out in your body, right? You don't even have reverse transcriptase unless you get the HIV virus, which is important because going back to these conspiracy theories about the COVID-19 vaccine, the COVID-19 vaccine is RNA, it's mRNA, so it does absolutely positively nothing to your DNA. The only way it could do anything to your DNA is if you had a reverse transcriptase. And it would have to be specific to the code in the virus, in the vaccine. But anyway, for the exam, again, if anything, you need to know the retrovirus goes from RNA to DNA, right? And the enzyme that does it is reverse transcriptase. Here's a picture of what it looks like. There's the envelope with the spike proteins on the outside. On the inside, you've got the protein that's boxing up the genes. In this case, those genes are, again, mRNA, but there's also an extra enzyme called reverse transcriptase. So that's what happens, right? The viral DNA comes in, reverse transcriptase does its job. It reads the code, And from that code, it makes a template strand. It makes DNA instead of RNA. And then that DNA gets double-stranded, and this is where it gets nasty. This is why HIV is so bad and so hard to deal with, is because that is then incorporated into your actual genome. It goes into your chromosomes, right? And then from there, your body, it just does the normal thing. It transcribes it, and it makes mRNA, then that comes out, and that's translated, and it makes a bunch of proteins that make up the virus, and those things are assembled, and then it leaves the cells and then goes on to infect new cells. I will say this, there's other viruses that do this too. It's not just HIV, but a lot of viruses actually insert themselves into your genome. And there's a lot of things in our human evolution that probably happened because of some ancient virus. It's actually quite interesting. I wish we had time to talk about it, but we don't. Any questions about this picture? All right. If you download the PowerPoint, you can watch the HIV reproductive cycle in action, a little animation of it. Your book gets pretty specific. I said earlier that HIV is so bad. One of the reasons it's so bad is because it attacks your immune system. And then right here, your book just gets very specific on how it attacks your immune system, but I'm not going to talk about it. We'll really quickly say that there are some drugs. Of course, this is old, but there's two different types of drugs that they use, and both of them interfere with the reproduction of the virus. So the first one basically inhibits the protease enzymes, which helps produce the HIV proteins. So that's an enzyme we didn't even talk about, but there's a drug that kind of keeps those enzymes from doing what they do. They are enzyme inhibitors, as you learned about when we learned about enzymes. And the second one inhibits the actual reverse transcriptase. And as you know, that's the thing that reads the code and makes the viral DNA, right? So if that doesn't work, then it's not going to work. Then the virus isn't going to work. The next slide is going to talk about this form and function. I think it's pretty interesting. None of you are chemists, right? But to your non-chemist eyes, wouldn't you agree that this thing looks a lot like this thing, right? They look so similar. They have the same shape, a lot of the same letters, right? And it's true, they are very similar. They have a very similar form, therefore it's a very similar function. What we're looking at here on the left is a normal T. Remember, there's A's, T's, C's, and G's. This is the T. You've seen it before. And what we have here is the OH. This is the part that gets covalently bonded to the phosphate. Remember the phospholipid, the sugar phosphate backbone? This is what has it covalently bond to the next nucleotide. Now, this drug gives something that looks a lot like a T, except it has a nitrogen here. So this thing cannot covalently bond to the next base, to the next nucleotide. So when your cells incorporate this into it when they're doing transcription, that's it. That's the end of the line because you can't attach anything to it. So that's how that works. Not that I'm going to ask you that for the exam, but are there any questions? All right. The last word for attendance is going to be prions because that's what we're about to talk about. And I'm going to talk about this very quickly. Most likely it won't be anything on the exam. You've probably heard of prions and just don't know you've heard of prions. So here we go. Very quickly. If anything, the only thing you need to know about prions, obviously they're infectious because everything we're talking about is infectious. They affect the brain. You need to know that, right? So they're infectious. They infect the brain. So everything we've been talking about so far has been nucleotides, right? It's either been RNA or DNA. This is different because it's a protein. Know that prions are infectious. They cause brain diseases. It's a protein. How does it happen? Basically, it's a misfolded protein, and every time it touches other proteins, then those become misfolded. But I probably won't even get that detailed on the exam. Just know these things, these first three bullet points right here. And there's more to talk about. Some diseases caused by prions, which I'm not going to ask you about. Scrappy and sheep, chronic wasting disease in deer and elk. If you guys hunt, you might know about that one. Mad cow disease, that's the one most people have heard of. Creutzfeldt-Jakob disease, that's a big one because that one affects humans. And you can look that up. Some people do have that. And it's a certain group of people that have it more than other people. And you can look it up if you want. Kuru was another interesting one. Again, that's a human one. And it's caused by prions. But the way it was spread was through cannibalism. So if you want to look that up for independent work, go for it. And that's it. Yeah, that actually is it. We finished it just in time. You know what? Lab will not be in person this week. I know I was saying that last week, but we'll just meet online. Yeah, that's it. So I'll see you guys either online for lab or on Wednesday.
Can you hear me? Hello? Hello? Can you hear me? Hello? Hello? I gotta ask him this question. Hello? Can you hear me?